Starting from the left, we have the preparation area where material and components are initially handled. Next is the changing room or gowning area, which ensures that personnel are properly gowned before entering controlled environments. Material pass through the material airlock to prevent contamination. Next is the aseptic corridor, which acts as a buffer zone, maintaining cleanliness and reducing contamination risk. Finally, we reach the aseptic filling room area on the right-hand side, which is the most critical zone where the actual product filling occurs under strict aseptic conditions. This layout is designed to ensure a unidirectional flow, minimize contamination, and comply with GMP requirements. Function performed in prefilled syringe production line. First, we start with the preparation of materials and components, where the syringe barrel, plunger, needles, and other parts are cleaned and sterilized. Next is aseptic filling, where the product is filled in clean room using specialized machines that maintain control over pressure and temperature, ensuring accurate dosing with minimal overfill. Then is component assembly, where all the sterile parts are assembled, often using automated equipment for precision. This is followed by a finishing, where the syringes are labeled, packaged, and inspected either visually or through automated system. Finally, testing and quality control is done. Check for sterility, particulates, and bioburden, ensuring that every single syringe meets safety and regulatory standards before release.
In process quality control, it plays a crucial role in ensuring the safety, efficacy, and quality of pharmaceutical products during the manufacturing. First, we have the identification test. This test is designed to confirm the presence of the intended active pharmaceutical ingredient, or API, in the product. It also helps differentiate the API from other structurally similar compounds that could cause confusion or contamination. The specific identification method used is always outlined in the USP monograph for each drug. Next is the assay test. This is a critical, stability indicating, and specific test that determines the strength or actual content of the drugs in the final product. This ensures the patient receives the correct dose as intended. Also, we have the impurity test. It is essential to check all the pharmaceutical products for impurities to make sure they fall within acceptable limits. Excess impurities can affect the drug safety, stability, and effectiveness. Moving on to the sterility test, as outlined in USP Chapter 71, this test ensures that parenteral products like injections and infusions are free from any wearable microorganisms. For sterile syringe, the contents should be expelled into separate membrane filter furnace or pooling vessels, and filtration must be performed immediately to maintain sterility. If the product has antimicrobial properties, the membrane filter should be rinsed at least 3 times using the sterile diluent. Make sure that each rinse passes completely through the membrane to remove any potential microbial residues that could interfere with the microbial detection. Next is bacterial endotoxin test. Based on USP 85, this test detects the endotoxins, which are toxic substances found in the outer membrane of gram-negative bacteria. This can cause the serious reaction if introduced into the bloodstream. There are 2 primary methods. First, the gel clot method. It is qualitative test. If the endotoxin are present, the solution will form a firm gel. So the second one is the turbidity metric method. It is quantitative. It uses a spectrophotometer to measure the cloudiness or turbidity of the sample, giving a precise endotoxin concentration. So this method is particularly useful when we need the exact levels of the regulatory compliance. Next, we have the volume in container test as described in USP 1. This test ensures that each syringe contains at least the labeled volume. The number of containers tested depends on their size. 1 container for volume more than 10 ml, 3 containers for between 3 to 10 ml, 5 containers for volume less than 3 ml. The entire contents are transferred to a third beaker, and volume is calculated by dividing the mass by density of the solution. The visual inspection test checks for visible defects in the product and packaging, such as cracks, chips, scratches, or particulate matter. This step is critical to ensuring both the product safety and integrity. In many facilities, an automatic visual inspection system is used to enhance the accuracy and efficiency. Lastly, the particulate matter test under USP 788 ensure injectable products are free from unwanted particles. There are 2 methods. First, the light obscuration test, which detects the particles by measuring the amount of light blocked as they pass through a sensor. The second one is the microscopic particle count test, where the particles are visually counted under a microscope.
In PICS guideline under General Information, we document system descriptions, which include preparations, revision, distributions, and archiving. This should also involve any change control and handling both electronic and paper documents. Next, laboratory documentation, which should include specifications, SOPs, test instructions, and analytical procedures. Then we should also ensure data traceability by documenting the procedures, record on receipt and usage of materials and standards. It also includes sample tracking and managing analytical raw data for full traceability. Lastly, electronic documentations or computerized systems are accepted as long as they meet the same requirements as paper documents. ICH Q7 guideline emphasizes the importance of document control to ensure data integrity and traceability. Section 6.1 covers documentation systems and specifications, where all paper and electronic records must be prepared, reviewed, and approved. In Section 6.2 covers equipment cleaning and use records, while Section 6.3 covers records for raw material, intermediates, and labeling and packaging materials to include batch number, test results, and supplier details. Next, Section 6.4 outlines master production instruction, which should contain detailed manufacturing steps. Section 6.5 covers batch production records, which must include materials used, yields, deviations, and signatures. In Section 6.6, laboratory control record must record all raw data and test method, while Section 6.7, the batch production records review, where should prepare document for all batch and lab records review before product release.
In a pre-filled syringe production facility, various stages of the process require specific staffing to ensure the efficiency, compliance, and product safety. The filling area typically needs 2 to 3 machine operators to handle the equipment, along with 1 supervisor to oversee performance and compliance and 1 maintenance technician to address any technical issues. For visual inspection, if it's manual, 4 to 6 inspectors are required, but if only automated, only 1 to 2 machine operators are needed, along with a supervisor to monitor the process. During the labeling and packaging stage, the staffing required depending on the level of automation. Automated process generally needs 2 to 3 machine operators, while manual tasks require 3 to 6 packers, all supervised by a manager, to ensure a proper workflow and documentation. In the quality control and assurance area, 1 to 2 QC analysts are needed for in-process testing, while sampling technicians collect and label samples, and QA officer review batch records and address any deviations. Lastly, maintaining a clean room standard is essential, particularly for Grade B and Grade A area. This is managed by monitoring technician and sanitation staff who ensure a sterile environment. In total, a medium-scale facility typically requires 2 to 28 staff members per shift for a single production line, though this number is variable depending on factors such as automation, bench size, and regulatory requirements. Proper manpower planning is critical to ensure the smooth operation and product safety throughout the entire production process.
This slide is about personal training and evaluation. An essential part of maintaining sterile products quality is ensuring that all the personnel are properly trained and evaluated. This includes demonstrating proficiency in the core skill required for aseptic processing. Staff must be trained not only in the theory, but also in hands-on techniques. One key area is proper garbing and hand hygiene. This is the first line of defense against the contamination. Competence in this step is assessed regularly to ensure the strict adherence to the protocols. In addition, aseptic manipulation techniques must be assessed through structural competency testing, ensuring each person can perform critical tasks under clean room conditions without compromising the sterility.
This slide presents a comprehensive overview of the personal hygiene and garbing procedure essential for maintaining aseptic condition in the production of pre-filled syringes. The process begins with a personal preparation which includes health screening to ensure with no staff member exhibiting symptoms of illness or open wound. All personnel must undergo appropriate training in aseptic technique and contamination control. Additionally, personal items such as jewelry, cosmetic, and nail polish must be removed and fingernails should be kept clean and trimmed. Following this, hand hygiene plays a critical role. Personnel are required to wash their hands thoroughly without no shedding antibacterial soap and warm water, paying special attention to the area beneath the fingernails. After drying with lint-free towel or using an air dryer, alcohol-based sanitizer should be applied. At all times, contact with non-sterile surface must be avoided. Garbing procedure follow a specific sequence to ensure sterility is maintained. This includes donning a hairnet on bed cover, face mask, eye protection, shoe cover, and sterile footwear, sterile glove or gown, and coverall. If necessary, sterile sleeve cover. The donning sequence must be carried out systemically, starting from the top of the body, proceeding to the downward, to prevent contamination of clean garment and areas. Equally important are the core garbing principle, minimizing movement and conversation, avoiding contact with the outer surface of sterile garment, and always position oneself appropriately relative to the clean room airflow. Any damaged or potentially contaminated clothing should be replaced immediately. In summary, each step in this hygiene and garbing protocol contributes to maintaining a contamination-free environment, ensuring the safety of the final product, and upholding the compliance to the GMP regulations. Based on guidelines from Pharma Guideline, cleaning and sanitizing in quality control, QC, and microbiology and production department should follow a structured frequency to ensure sterile condition. Ceilings are to be cleaned monthly using a vacuum cleaner or moist duster. High contact surface like a door, doorknob, switches, and tables are cleaned weekly using potable and finally purified water. Floor require more frequent attention, being cleaned twice daily using a rotation combination of 2% Savlon and 1% Dettol in the QC lab. Glass surface are cleaned weekly with 5% Dettol or Savlon solution, while walls follow a similar weekly schedule with the same disinfectant rotation used for the floor. The laminar air floor units are sanitized before and after work using 70% IPA as mandated by the microbiology lab. This systematic approach ensures microbiological control and compliance with good laboratory and manufacturing practices.
Proper cleaning and disinfecting of the primary engineering control, or PEC, is important to maintaining a sterile compounding environment. Standard procedures begin by using a sterile, low-lean wiper with a treatable solution such as sterile water for injection or irrigation to remove any visible particles or residue. Next, all internal surfaces and equipment inside the PEC are wiped down using a sterile, low-lean wiper moistened with a sterile cleaning agent. This is immediately followed by an EPA, a registered sterile disinfectant. It is important to allow the disinfectant to remain on the surfaces for a required contact time, as recommended by the manufacturer, to ensure full effectiveness. Finally, a wipe down is done using the sterile 70% isopropyl alcohol to remove any residual disinfectant and provide an added layer of microbial control. Before starting any compounding, we must confirm that all the surfaces are completely dry.
Procedures to apply a sporicidal disinfectant in the PEC. First, remove any visible debris using sterile water and a low-lean wiper. Next, apply the EPA-registered sporicidal agent to all interior surface and under the broad tray. Measure the contact time specified by the manufacturers is min. Then apply sterile 70% IPA to all surfaces. Finally, let everything dry completely before starting compounding.
In the production of prefill syringes, typically use sporicidal agent to eliminate resistant bacterial spores and ensure microbial control. Sterile isopropyl alcohol (IPA) is a broad-spectrum disinfectant that acts quickly on surface and equipment, especially in aseptic zone. In addition, WFI water for ejection quality water is used for diluting, rinsing, and residue removal, ensuring no contaminant compromise the sterile environment. Together, these agents form the foundation of an effective contamination control strategy within clean room and classified areas. Cleaning supplies used in the prefill syringes production area must meet strict standards to preserve sterility integrity. All material must be low, lean, and sterile, especially when used inside the primary engineering control PEC. Tool handles require disinfection before PEC use, and disposable items like wipes and mop heads are preferred to minimize cross-contamination. Being discarded after each use, when reusable tools are employed, they must be made of cleanable, dedicated material and disinfect both before and after each use. Disposable methods should prevent the spread of contaminant into the air. No shaking or agitating near sterile zones. Lastly, sterile supply may be only reused, but if only guided by SOP or manufacturer instruction, ensuring safety and regulatory compliance.
This slide covers the clean room classification, which is based on the maximum allowable particle count per cubic meter of air, according to the standards like ISO 146441. In syringe production, clean rooms typically range from ISO Class 5 to Class 8, depending on how critical the process is and the level of cleanliness required. ISO Class 5 is the cleanest, ideal for aseptic processes like syringe filling. ISO Class 6 allows the slightly more particles but still maintain the strict control. ISO Class 7 and 8 have the highest permissible levels used for less critical tasks, such as assembly and packaging. Next, we have air filtration systems, which are essential for keeping the air in production areas clean and contaminant-free. HEPA filters remove 99.97% of particles that are 0.3 micrometers or larger. ALPA filters are even more efficient, can remove 99.9995% of particles as small as 0.12 micrometers. The pre-filters and fine filters help by trapping the larger particles before the air reach the main filters. Terminal HEPA or ALPA units are installed in critical areas to reduce the contamination risk. Lastly, the HVAC systems control the temperature, humidity, and airflow to maintain the optimal conditions for sterile production. Then, maintaining proper airflow control is essential to prevent contamination in the clean rooms and syringe production area. Directional airflow use positive pressure to keep clean air in, and negative pressure in hazardous areas to keep contaminants out. Air filtration using HEPA and ALPA filters remove the airborne particles for high air quality. Laminar airflow provides a smooth, consistent flow of clean air across the workspace and minimizing the contamination. The air exchange rate is regulated by the HVAC systems to keep the air fresh and clean. And finally, airflow monitoring and control systems constantly track and adjust the air movement in real time to ensure the environment stays within the safe limits. Temperature and humidity control. So maintaining a proper temperature and humidity is essential in syringe production areas to ensure product quality, manufacturing integrity, and sterility. Temperature is typically controlled between 18 to 25 Celsius with stricter regulation in clean rooms to protect the sterility and the product integrity. Humidity is maintained between 30% to 70% to avoid the microbial growth and product degradation at the high humidity levels. So at low humidity, the control is crucial to prevent the electrostatic discharge and attraction of the particles that can contaminate the product. The surface disinfections and cleaning. So regular cleaning and disinfecting of surfaces and equipment can prevent the microbial contamination. The clean room compatible disinfectants and standardized cleaning protocols maintain the sterility. Then the personnel guiding and training. The strict gowning procedures and throughout training in aseptic techniques and hygiene are essential to minimize the contamination risk and maintain the clean room integrity. Finally, the control access. Only authorized personnel can enter the syringe production areas. Access points like airlocks and air showers can help to reduce the contamination transfer. So that's all from our group. Thank you.